So this section is about cell signaling, how cells communicate with one another. It's not an entire chapter in your textbook, it's really just a section of a chapter. So first, let's talk about why cells would need to signal one another. So in a multicellular organism, it's fairly obvious that, for example, just for you to move, you need to coordinate what you see, sending messages to your brain, which sends messages out to your arms and legs. And then all of those cells in your muscles need to coordinate with one another, and that allows you to move. In single-celled organisms, we also see cell communication in many cases. Organisms can form these things called biofilms, where they work together and they protect themselves from invaders. We can see cells tell other cells to slow down their reproduction because it's getting too crowded. So even single-celled organisms can send chemical signals to identify friends and foes, or to tell the group to reproduce or not reproduce. So the simplest type of cell signaling would be direct contact. Gap junctions we've already talked about are plasmodesmata and plant cells, literally connect one cell to another, and cytoplasm can freely flow between them. Additionally, we can have a situation where a cell releases chemicals to stimulate all cells in a particular area, such as growth factors. So another example of this would be, for example, if you've ever slammed your finger in the door and that really hurts. You suddenly will feel that your finger gets hot. It starts to swell. What's going on there? Well, a cell signal's going out to all cells in the area saying, hey, let's raise the temperature to make this uninhabitable for bacteria. Let's open up the blood vessels and let white blood cells in case there's an infection that's going to set in. Let's tell the cells in the area to start repairing, start a blood clot, stop blood flow, make new cells to replace the cells that have been damaged. So that would be this type of signaling. Sometimes this is called paracrine signaling, and this is a picture of it, where one cell signals a whole bunch of cells in an area. A second kind of signaling is what's called synaptic signaling. Now, we don't talk about the nervous system in AP Bio anymore. It's not considered part of the AP exam curriculum. However, they could still ask you some questions related to this. So this is what a neuron looks like. It's a really long, skinny cell. This is called an axon. Again, you wouldn't be tested on these vocabulary words. And these are dendrites over here. And so, like, if you touch a hot stove, for example, these dendrites would be stimulated, and this would cause sodium channels to open, and that disrupts the electrochemical gradient that you have. And there's these gates that cause sort of a domino effect, and the nerve impulse travels until it gets to the end. The thing is, that nerve impulse can't jump from the tip of this neuron to the spinal cord if this was to send a message to the brain, or if this was a message coming from the brain, it cannot make a jump here to the muscle cell. So there's a gap here called the synapse, which is why this is called synaptic signaling. And at that synapse, chemicals called neurotransmitters get released, and they only signal just this next cell that they sort of fit right into. And so this is synaptic signaling. Sometimes neurotoxins like snake venom or Botox, things like that, might disrupt this type of signal and cause you to be paralyzed in a particular area because the neurotransmitters either don't get released or these receptors to pick up their signal are blocked, something like that. So this is what synaptic signaling looks like. So again, it's still localized because this neuron is very closely connected to this receptor target cell, whatever this might be, a muscle, your spinal cord, your brain, another neuron, whatever. And so this signal only goes out to this one specific cell. The last kind of signaling, which we're really going to focus on, is long-distance signaling, which is what your endocrine system does. So this is where you make hormones, or a more general term for a chemical that does long-distance signaling is a ligand. They use this word a lot on the AP exam. This is where one cell in your body may produce this hormone or a ligand, and then it travels all the way across your body through your bloodstream to some other cell in another location. A lot of times this is for the purposes of negative feedback, maintaining homeostasis. For example, if your blood sugar gets too high, you release insulin to lower it. If it gets too low, you release glucagon to raise it, and the idea is that you have this balance that's maintained. So this is long distance signaling like your endocrine system. So this is where we're going to focus. So there are 3 stages in this cell signaling, in this particular long distance kind of signaling. The first one is reception. The second one is called transduction, and the last one is the response. The transduction step is really the complicated one. And it looks something like this. So this is a receptor. In this case, the receptor is in the cell membrane. So the ligand, the signaling molecule, this could be a hormone, for example, it's bonding to the receptor. Notice that this molecule never actually enters the cell. The receptor, some kind of a change happens that it sends a signal to these relay molecules. That's going to be our transduction step. And then something's going to be activated as the cell's response to this. Now, I wanted to point out, notice how this receptor is on the cell membrane. Not all receptors are on the membrane. And this goes back to our cell membrane chapter where we talked about what can and cannot pass through the membrane. So water-soluble ligands, hormones, protein hormones, for example, are not able to pass through the phospholipid bilayer because it only likes, remember, small lipid-soluble things. So a hormone that's a protein hormone would bond to a receptor on the membrane like this picture. On the other hand, a steroid hormone, which is lipid-soluble, would pass right through the phospholipid bilayer, and it would still need a receptor, but the receptor would be intracellular, meaning within the cytoplasm. So here's a picture of that. So this is the hormone. In this case, they're showing testosterone as a generalized steroid hormone. It bonds to a receptor inside. That receptor, then that complex can take the message to the nucleus where some kind of a response happens. So it is very possible that they could show you a picture and show you either a hormone binding to a receptor on the surface or that it goes right through the membrane and bonds to a receptor inside and ask you a question that would require you to know whether it was a protein hormone, which would need a surface receptor, or a steroid, which could pass right through. Sometimes you have something called a coupled receptor. You're going to see this in the quiz that I assigned to you to take. So in that case, and it's not something you have to memorize, but you should recognize, notice what happens is that the ligand bonds to this original molecule. In this case, this is called a G protein. When this bonds, it releases this protein, and it goes and bonds to a second protein in the membrane, and that then initiates a response. So it's called a coupled receptor because there's sort of 2 parts to that reception portion. All right, so let's talk about the second step. So the second step is called transduction, and its goal is to amplify the signal. Sort of like when a video goes viral, right? One person makes the video, and maybe they share it with a couple people. Man, those people really like it, so they share it with a whole bunch of people, and each of those people share it, and then the next thing you know, you've got 5,000 or 5 million views, right? And it all depends on how much of that sharing goes on. Well, it turns out your cell can control that. This step is highly controlled, so that instead of, and I gave the analogy here, you can, in your house, you may have some light switches that are either on or off. But that's not very convenient for everything. For example, if you're driving your car, if your car only had stop and go 60 miles an hour, that would be really difficult to control. On the other hand, if you can speed up and go 10 or 15 or 60 or 90, you can go as fast or slow as you want. Same kind of idea here. Your cell can either have a big amplification step here or a small one. So it's a way of better controlling the cell's response to a ligand. All right, so this involves something sometimes called a cascade event, and specifically it involves something called second messengers. In other words, the protein hormone didn't ever enter the cell. This would be my receptor, and it triggers these molecules in between, and these are your second messengers. They're taking the message because they're not actually carrying out the response. They're just amplifying it, carrying it to this final goal place, maybe the DNA, to activate it to make a protein or turn on an enzyme or open a channel or something. But the final response is gotten to by this sort of domino effect of this person tells 2 people and they tell 3 people, and it goes on from there. There are a lot of different second messengers. 2 common ones you might see would be cyclic AMP or for protein kinases. ACE tells you this is an enzyme, and kinases in general, what they do is they add phosphate to things, or phosphorylate. And the reason that that's important is because adding phosphate to proteins tends to activate them. We learned that in the sodium-potassium pump. Remember, the 3 sodiums bound to the protein. When ATP dropped off a phosphate, that's what popped that protein into a new shape and transported the sodiums across. And we actually see that in a lot of cases. That's actually how ATP powers reactions, is that phosphate, when it gets passed to something, it activates it. So this is an example of something like that. You can see how complicated these can look. On the AP exam, you would typically be given a picture with several steps, and then they would say, you know, this step is inhibited, or this gets stuck, or this is the wrong shape, or this part you make too much of. And then they'd want to know which logical conclusion you could reach from that. So this is our original receptor. All of this is our second messengers. So this kinase gets activated by this molecule released here, which activates this kinase, which triggers ATP to break and add a phosphate to this, which then triggers another ATP to add a phosphate to this, and there's a whole complicated response. The final response can be multiple things. It depends on the hormone. It could be to make an enzyme, turn on DNA, turn on gene to make an enzyme or a protein of some kind. It could cause an ion channel to open. It can activate an inactive enzyme. It could cause you to break something down. There's lots of things it could be. So here's a summary. So this is our reception, this is our transduction, and then this is our response, in this case turning on a gene.
Cell-to-cell signaling, with molecules carrying signals from sending cells to receiving or target cells, is a key mechanism in the coordination of cellular activities. In most cases, a signal molecule acts by binding to a receptor protein in the plasma membrane of the target cell and activating a series of changes, a signal transduction pathway, that triggers a specific response within the cell. A signal molecule binds to a specific receptor in the plasma membrane of the target cell. This activates a series of relay proteins, which in turn activates a transcription factor. The activated transcription factor attaches to the DNA and triggers transcription of a specific gene. The mRNA message is translated into a new protein, which changes the structure or function of the receiving cell.
All right, so that kind of gives you an overview of what I'm talking about. This is just a generalized one. Here's a more specific one. So in this case, the hormone adrenaline or epinephrine, what it does is it binds to a receptor. It activates a second messenger called IP3. That causes calcium ions to be released. This is in your liver cells. Those calcium ions trigger an enzyme called glycogen phosphorylase. That breaks glycogen into glucose, and then the glucose can go into your bloodstream to go to all your cells for the fight or flight response. So this would be an example. You don't have to memorize any specific examples. You just are going to have to understand how to read these from diagrams. All right, and then to summarize, one last thing about feedback. So negative feedback is very common in your body. This is, again, maintaining homeostasis. Positive feedback, what that is, that's where the product of a pathway causes the pathway's activity to increase. Normally, this would not normally happen in your body. If getting hot caused your body to make you hotter, that would not be a good thing, right? But childbirth and blood clotting are 2 examples. Let me just go through childbirth for you real quick. So when the baby's head starts pushing on the cervix here, that stimulates you to make oxytocin. Oxytocin stimulates contractions. Well, contractions cause the uterus to push the baby against this cervix even harder, which is going to make more oxytocin, which is going to make more contractions, and this just builds up to this climax event where you give birth. So this would be an example of positive feedback because the pushing on the cervix causes more of a hormone that increases the pushing on the cervix. And it always culminates in something. Blood clotting is another example of this in your body. All right, so that's kind of an overview of cell communication. I don't think you're going to really get the gist of this until you try some practice questions in that quiz that I gave you. But I find that these are not difficult, but they're time-consuming AP questions to answer.